So we'll go through tutorial 7 now. So that's going to be covering biotechnology. So we should get through this pretty quickly, and then at the end we'll do some revision questions, which are kind of similar to the stuff that will be coming up in your mid-semester exam. So first of all, we'll cover therapeutic proteins. So there's a number of diseases where the proteins that are necessary for healthy functioning is inadequately produced. So examples are insulin. So if you can't produce insulin, you've got type 1 diabetes. And if you're deficient in blood clotting factors, you've got hemophilia. And you could also have a deficiency in growth hormone. Okay, so remember that those are all proteins. So yeah, I don't know what I was going to say about that. Yeah, so these diseases can be treated by administering the protein, and that's going to usually be by injection. Okay. So why would you need to inject the protein? Why can't you just take it by mouth? Well, yeah, that's one advantage of injection, but usually when you're taking a drug, people don't like injecting themselves, so it's better if you can just take it by mouth. But a protein is a long chain of amino acids, right? So if you have a long chain of amino acids and then you swallow it down your throat and then it goes into your stomach, what's in your stomach and what's in your intestine? What sort of enzymes do you have? You've got digestion enzymes, you've got proteases, and proteases are going to cleave the peptide bonds, and they're going to break up that protein, right? And so if you, say for insulin, for example, it's about 50 amino acids long, and if you chop it up so it can be absorbed into your body, you've absorbed those amino acids, but now they're all in like they're only 1 or 2 amino acids long, so you've lost that function completely. Okay, so that's why you need to inject the protein so it bypasses, it doesn't go through your intestines, it'll be absorbed into your bloodstream directly. Okay, so that's just one thing to think about when you're trying to replace these proteins. They're going to have to be injected. And in the past, therapeutic proteins were extracted from animals. And so the proteins that were extracted from animals, for example, insulin, they had slightly different sequences of amino acids. So what do we call the structure? Is it primary, secondary, or tertiary? The sequence of amino acids. Yeah, primary. So they'll have slightly different primary structures, and because they're slightly different, the immune system of the body is going to see that protein and it's not going to recognize it. It's going to think it's a foreign substance, which it is, and it's going to attack it, and so you'll get an allergic response. And proteins are also extracted from humans, but this was also associated with infection risk. So in the 80s, a lot of people with hemophilia had to replace their blood clotting factors from other people, and they ended up getting HIV and hepatitis because a lot of these products were contaminated. So recombinant proteins are proteins that have been produced using recombinant DNA technology. So recombinant DNA, so recombining. So you've combined DNA from different species in the lab, and the gene that codes for the desired protein is inserted into a plasmid. and the protein is then produced in the cell system. So recombinant proteins have the same primary structure as the normal protein, and so they don't cause allergic reactions. So if the protein has the same primary structure, it's going to have the same secondary structure, it's going to have the same tertiary structure. So to the immune cell, it looks exactly the same as a protein that would normally be present in your body, right? So then it's not going to attack it, and you won't get an allergic reaction. And the advantage of using recombinant proteins is that because they've been created in the lab, they have a much lower risk of being contaminated. And so these recombinant proteins are used to treat patients with a disease by replacing the protein that may be non-functional or not produced, but they are not a permanent cure, right? So you have to, if you have a type 1 diabetic, that person has to keep injecting insulin for the rest of their lives, because they're not actually producing the protein. It has to be replaced from outside. So keep that in mind. And that diagram at the bottom there just shows some recombinant proteins that are being used to treat diseases. So you can see there's quite a few. There are quite a few diseases that are treated with recombinant proteins, and they've made a huge difference in the medical field. So plasmids are circular pieces of, are they single or double-stranded? Yeah, so they're double-stranded. Yeah, don't be afraid to be wrong. Like, I'd rather that you guys shout something out and be wrong than like not say anything just because you're scared of being wrong. Yeah. So well done. So plasmids are circular pieces of double-stranded. Is it RNA or DNA? Yep. So it's DNA, and that's usually found in bacteria. So they're used as vectors, and a vector is something that's constructed to introduce a gene for expression into a cell. So a vector could be a plasmid, or it could be a virus. So basically anything that's used to insert a gene into a cell. So if you look at that diagram there, you'll see that you need to have two things. You need the DNA sequence or the gene that you want to insert, and you also need the plasmid, and you need to open up that plasmid and then insert the gene. And the enzymes involved in that is the restriction enzymes and the DNA ligase. Okay, so the gene that encodes the protein required is inserted into the plasmid using restriction enzymes and DNA ligase. The DNA sequence containing the gene will have been modified at each end so that there will be a sticky end. So the sticky end is a single strand overhang of DNA nucleotides, and they'll be present at each end that can pair with a complementary sticky end on the plasmid. And you'll also need to have an origin of replication on the plasmid. So the origin of replication is the site where bacterial DNA polymerase will bind, and once DNA polymerase binds, it's going to start replicating that plasmid, and so you can make many more copies of that plasmid. So if you didn't have that origin of replication, the plasmid wouldn't be able to replicate, and so the plasmid would be diluted out with each bacterial cell division. So you've got your, let's say you've got your, this is your bacterial cell, and let's say you start off with 4 plasmids. So this plasmid does not contain an origin of replication. So this is with no origin of replication. So then these plasmids can't replicate. So when the bacterial cell divides, it's going to be diluted out. And then so on and so forth. When this cell divides, each cell will only have 1 plasmid. And then when this cell divides, one of the cells is not going to have a plasmid. And then you've kind of defeated the purpose, because if it doesn't contain the plasmid, which has the gene that you want to express, then that cell isn't going to produce your protein, right? So you need to have the origin of replication on it. Let's say it's this thing here. So let's say you just start off with 2 plasmids in the cell. If you have an origin of replication, then you can replicate those plasmids before you divide, and then so then each cell will get 2 plasmids, and then this cell will replicate those plasmids. So you'll have 4 plasmids in each of the cells. And then each cell will inherit 2 plasmids, but they'll be able to replicate those plasmids. And so the number of plasmids inside each cell is not going to get diluted out. So that's why you need the origin of replication. And then you also need to have a selectable marker. So usually the selectable marker is going to be an antibiotic resistance gene. And so this allows for the selection of cells that have taken up the plasmid. So let's say when you're trying to make these recombinant proteins, you're going to have all these bacterial cells, and you're going to have the plasmids, which are outside. And so you want the bacteria to take up these plasmids, but some bacterial cells won't take up the plasmids. Okay, so what you do is you're going to put a selectable marker, which is an antibiotic resistance gene. So all the plasmids will have an antibiotic resistance gene, and so from this, I'll draw it a bit larger so you can see it better. So here's 2 bacterial cells. This cell has taken up the plasmid, while this cell has not taken up the plasmid. So this plasmid contained, So this plasmid has that antibiotic resistance gene, and that antibiotic resistance gene is going to be transcribed, right? So that's transcription. And you've got the ribosome of the bacterial cell, which is going to translate that mRNA, and you'll get. So this is the this is an enzyme that will inactivate antibiotic. So you've got this enzyme here that's going to inactivate the antibiotic, and so only the bacterial cell that has taken up the plasmid is going to have that enzyme. So now, if you introduce an antibiotic into the mixture, the reason that you want to select is because if you don't select for them, both of these bacterial cells are going to grow, and so this bacterial cell here will also have the gene that you want to produce, right? And so this one's also going to be producing the protein that you want, whereas this bacterial cell won't be. And if you just let both of them grow, this one's going to probably grow faster because it doesn't have to produce these proteins, and so you want to kill these ones off so that you only grow these cells. Okay, and so the way you do that is to introduce antibiotic into the mixture, and the antibiotic will diffuse into the cell. And when the antibiotic diffuses into this cell, which doesn't have the enzyme that's going to inactivate the protein, inactivate the antibiotic, this one's going to die, whereas this one can inactivate the antibiotic, so it's going to live and replicate, and you'll just get more and more bacterial cells that contain your plasmid, and they're going to produce your proteins. The protein that you want. Okay. Right. So then the promoter must be specific to the cell system that is being used to manufacture the protein. So for example, if you're using a bacterial cell system to express a human gene, such as insulin, the promoter must be a bacterial promoter, so that bacterial RNA polymerase can bind. Okay. So I'll just draw a diagram quickly. So you have this is a bacterial cell, and here's your so let's say you have inside your bacterial cell, you have two plasmids, and on plasmid number 1, you have this is your, let's say this is a mammalian, no, let's say this is a human promoter. And then this is your insulin. This is your human insulin gene. And then so this is plasmid 1, this is plasmid 2. On plasmid 2, you've got bacterial promoter, and you've got the human insulin gene. Okay, so you've got inside your bacterial cell, the bacterial cell is going to produce, this is going to be bacterial, bacterial RNA polymerase. Okay, so if you have a human promoter on your plasmid, this bacterial RNA polymerase is not going to recognize the human promoter because this human promoter has a different DNA sequence to the bacterial promoter. But then, so you've got this bacterial RNA polymerase, but then if you have a bacterial promoter on your plasmid, it's going to recognize it, and so it's going to bind, and once it binds, it's going to start transcription. And so some people ask me, well, how come you can transcribe a human gene? What's important is the binding of that bacterial RNA polymerase. Once it binds, it'll just start transcribing. It doesn't matter whether it's a human gene or a bacterial gene or like a yeast gene. Once it starts transcribing, it doesn't really care what it is. It's just going to copy it. But it's the binding. So it's the DNA sequence of that promoter that's important. Okay, so if you're putting a plasmid in a bacterial cell, you need to have a bacterial promoter. And so if this was a mammalian cell, you would need to have a mammalian promoter. Okay, and also if you wanted to, let's say this was you wanted to you only wanted to produce the protein in a milk producing cell, then this promoter would have to be milk producing cell specific promoter. So if you only wanted to produce in a cell that produces milk, like in a transgenic animal, then yeah, you need to have a special promoter for that. Okay. So we'll move on to the next page, which is creating a recombinant protein. So the steps involved, the PCR. So just like when you're doing genetic testing, you can't just make one plasmid because these plasmids are absolutely tiny, so you can't even see it or you can barely see it if you have a microscope. Initially you want to create lots of copies of your plasmid, and so that means that you need to have lots of copies of your your gene as well. And the DNA sequence has to be flanked at each end by sites that can be recognized by the restriction enzyme used to cut open the plasmid. So you might have to modify the DNA sequence on either end of the gene to make sure that it has a site that can be recognized by the restriction enzyme. So you might, and then the reason that you do that is so that the sticky ends of the plasmid and the gene insert are complementary and they're going to bind to each other. And then even once they bind to each other, they're still not 100% connected, so you need to have the DNA ligase enzyme to seal them together. So once you have sufficient numbers of plasmids containing the gene, they must be taken up by the cell by a process called transformation. So transformation can be induced by heat or electrical shock. So you're going to select for the cells that have taken up the plasmid by applying an antibiotic. You allow the bacteria or the mammalian cell culture to grow. And since they have a promoter which drives the expression of the gene, the protein will be produced by the cells and extract and purify the protein. Okay. So then we'll cover the main features of the prokaryotic system, which is the bacterial system. And the most common bacteria that's used for this is E. coli or Escherichia coli. So bacteria is most commonly used to produce recombinant proteins. There's a few reasons for this, as we'll go through later. So the plasmid is taken up by transformation, and the bacterial cells, so they can be selected for. So what type of promoter do we need? So we need a bacterial promoter, since the bacterial RNA polymerase is different to eukaryotic RNA polymerase. So the pros of a bacterial system is that it's very low cost, so it doesn't cost a lot to grow bacteria. And then you can get a lot of proteins from bacteria. So you'll all have heard that bacteria can divide like once every 20 minutes or so they can divide really fast. And as long as you provide the nutrients for the bacteria to grow, they'll grow really fast and produce a lot of protein for you. And they're free from human pathogens like HIV and hepatitis and Creutzfeldt-Jakob disease, things like that. But the cons, so they can't carry out post-translational modifications or PTMs, such as glycosylation or gamma carboxylation. And because the intracellular environment of a bacteria is different from the intracellular environment of a human cell, that's going to affect the folding of the protein, so they're often going to be not folded properly. But the reason that you can still produce insulin in bacterial cells is because insulin is quite a simple protein. It's only about 50 amino acids long, whereas some proteins can be hundreds or thousands of amino acids long. So the longer they are, the more complex their folding is going to be. That's why you can produce insulin with bacteria, but a lot of the other proteins you can't. So insulin is produced as two separate polypeptide chains. You've got the A chain and the B chain. So one group of bacteria is going to produce your A chain, So you've got this group of bacterial cells producing A chain, and this group of bacteria producing the B chain, and then you're going to extract the protein from both groups of bacteria. Okay. So you're going to extract, they're going to be made separately, and then you're going to, once they've been extracted from the bacteria, you're going to mix them together, and then you need to let them, so what sort of bonds are going to form between the A chains and the B chains? Yep, so disulfide bonds. So in order to form disulfide bonds, they form by oxidation. So you need to mix the A and the B chains under oxidative conditions so that the disulfide bonds can form, and then you'll have your functional insulin, which can then be used to treat diabetes. So insulin can also be produced by other recombinant systems, such as mammalian cells and transgenic animals. I say this here because in one of the previous questions in previous years, one of the questions was insulin can be produced by, and the options were A, bacteria, B, mammalian cells, C, transgenic animals, and then D, all of the above. And so the answer is all of the above, because they can all make it, but it's kind of pointless to make such a simple protein with mammalian cells or transgenic animals, because why would you do something that can be done cheaply in a more expensive way? So it is possible, but for realistic purposes, you'd only use bacterial cells. So bacterial cells cannot remove introns. So the gene that you insert has to be free of introns. So you need to create cDNA. So cDNA is complementary DNA. And if you look at that diagram on the right side, that'll show you how you're making that complementary DNA. So basically, you take the mRNA that's already had the introns removed from a eukaryotic cell, and then you mix it with reverse transcriptase. So you're going to create a DNA transcript from the mRNA, and then that's your cDNA, which doesn't have any introns in it. Okay. And then eukaryotic cells. So the most common cells that we use is Chinese hamster ovary cells, which are used to produce EPO. And so these aren't actual hamsters that they use in the lab, so they haven't got hamsters locked up or anything. They just took the cells originally from the hamsters. And so the protein has to be glycosylated. So a glycosylation is, if you have the protein here, you're just adding, so glyco means sugar, right? So glycosylation means adding a sugar. So you're just adding a sugar to that protein. And because bacterial cell systems can't do glycosylation, you have to use a mammalian cell system. And so if you're using a mammalian cell system, you need a mammalian promoter. And so the pros are the proteins are usually going to be folded properly, and you can carry out most of the post-translational modifications. The cons are that it's more expensive than bacterial systems, even though it's cheaper than transgenic animals. And mammalian cells divide more slowly than bacterial cells, so it's going to take more time, and you're also going to not get as much yield from these systems. And there are still some post-translational modifications, like gamma carboxylation, that you can't do inside a single cell that's been isolated. So then transgenic animals, so usually it's going to be cows and goats that can be genetically engineered to produce proteins in their milk. And the good thing about milk, about making them produce proteins in the milk, is that you can just purify the milk instead of having to kill the animal. And then so a transgenic plasmid will contain a milk-specific promoter to drive transcription of the target gene only in cells that are involved in milk production. So the way they do this is to have a promoter region that contains binding sites for transcription activators that are normally present only in milk-producing cells. Okay. So that even though the gene's present in all of the transgenic animals' other cells, it won't be expressed. So the example that you got given was antithrombin. So that's a protein that requires gamma carboxylation. And the pros is that because you've got a whole animal, and cows and goats can usually produce a lot of milk, you can get a lot of yield, and you can perform all your post-translation modifications. But the cons is that you've got a high cost, so it's very hard and difficult to make a transgenic animal. And in New Zealand, I don't think you can even do any sort of transgenic stuff on animals. So there's lots of regulations and ethical issues. But on that note, I think, because in the past they had to extract growth hormone from like dead people, so they had to extract growth hormone from the pituitaries of dead people, and if you're deficient in growth hormone, you like end up being quite short, and there's a lot of other problems as well. But now I think if you have one cow, it can, or like maybe it's like 10 cows or so, like that's enough cows to produce enough growth hormone to treat everyone in the world with growth hormone deficiency. So that's just an idea of how useful it can be. Whereas before, it was quite hard to get enough growth hormone from dead people. And also there was the risk of infection as well. So in gene therapy, a functional copy of the gene is delivered into the patient's cells so that the patient's own cells can be used to produce the protein that is deficient or non-functional. Viral vectors are used to deliver the plasmid containing the target gene into the patient's cells. So remember, a virus can insert its DNA into your nucleus. So that's why viruses are used. But you wouldn't want to infect these people with a virus that's going to replicate and cause disease. So the virus has been modified so that it's not going to replicate, so it's not going to harm that person. And it is a more permanent form of treatment because you've introduced the gene which can produce the functional protein into that person's cell, so that that cell can now keep producing the protein. And so the key thing to note here is that the patient's defective gene is unchanged. It's still in that, it's still defective, and it's still in that person's body. You've just delivered a replacement. So the pros is that it's permanent or semi-permanent, and you don't need to constantly administer the protein. And the cons are that it's expensive, and it's only effective for monogenic diseases. So monogenic means one gene. So if you have a disease where multiple genes are involved, it's going to be a lot harder to treat with gene therapy. And you can also get an immune response to the virus when you administer the virus. That gene, your body can respond to the virus. And you can have problems if the gene is inserted in the wrong place in the patient's DNA. So because the virus is going to insert the gene randomly into your DNA, if it inserts it in the middle of a gene, it could break that gene, and then you're not going to produce that protein, or it could cause a mutation, and it could lead to cancer. So there were a few issues where they did like clinical trials, and then some of the people ended up developing cancer, and like one person died from like an immune response to the virus. So it can have like, you need to be careful with all this stuff, but there's a lot of potential benefit. And then gene editing, so it's using an enzyme called CRISPR-Cas, and this is used to edit the patient's defective gene. So the CRISPR-Cas is an enzyme that's present in bacteria to defend itself against viruses. So what the bacterial cell will do is, when the virus comes into the cell, it'll bind to the virus DNA and change it so that now it's not effective anymore. And it just involves breaking the DNA strands and then like editing the genome. So the key difference here is that compared to gene therapy is that the defective gene is fixed. So that's the key difference. So we'll just go over a few revision questions now. So number one, what type of amino acid is this, and where would you be most likely to find it? So who wants to answer that one? So is it polar or non-polar? Yep. So it's polar. So it's a polar acidic. So polar acidic. So you can see that it's got so this is the amino acid. So this is so this here, this is the alpha carboxyl group. This here is the alpha amino group. And so this here, this carbon here, is the alpha amino. This is the alpha carbon. Okay. So you need to be able to recognize which one the alpha carbon is. So it's the one that has an amino group and the carboxyl group attached to it. And then, so So that means that this here is the side chain, and the side chain has a carboxyl group on it. And so under normal circumstances, at a pH of 7, it's going to be deprotonated, right? And so it's going to be negatively charged. So that brings us, so I'll answer question 1 first. So it's a polar acidic and it's negatively charged. It's most likely found on the surface of the protein since it's hydrophilic. But it's still possible to find it in a hydrophobic core. So if you remember, so this amino acid here is this amino acid here is aspartate. And if you remember in the enzyme chymotrypsin, it's got a aspartate residue in the hydrophobic core of the protein because it's part of the active site. So it's not always going to be on the surface of a protein, but if you were to guess, you'd say it's most likely going to be on the surface of the protein because it's going to want to be next to all the water, which is on the outside. And so question number 2, what would the net charge be on the free amino acid shown above at pH 7? So if it's a free amino acid, it just means that it's not part of a protein. It's just free like it is here. So then at a pH of 7, this carboxyl group would be deprotonated, so it'd be negatively charged. This amino group is going to accept a proton, so it'd become NH3, and it'd be, this amino group would be positively charged, and this carboxyl group would be deprotonated. So you would have, this would be negatively charged. So you've got 2 negative charges and 1 positive charge. So that's a net charge of -1. And then, what would the net charge be if the amino acid was at the N-terminus of a protein? So I'm just going to draw out a tripeptide. So I'm going to draw out 3 amino acids connected together. So you've got the N, the nitrogen, the alpha carbon, carboxyl carbon, nitrogen, alpha carbon, carboxyl carbon, nitrogen, alpha carbon, carboxyl carbon. So you've got the N-terminus here. So at a pH of 7, this N-terminus will be positively charged. And so let's say this one here, so this one is the, it's on the end. So this R group will be our aspartate. So this carboxyl group will have lost its proton, so it'll be negatively charged. And you'll have the carboxyl carbon, the R group. So remember, the R group goes on the opposite side to reduce steric hindrance. Okay, so what's the net charge of just the aspartic residue? Yeah, it's going to be 0 because at the N-terminus. And this carboxyl carbon here is involved in the peptide bond, so it's not going to be charged. So the net charge is going to be 0 or neutral. But what about if it was on the C-terminus? if it's on the C terminus, now you've only got 2 negative charges, and you haven't got a positive charge because this nitrogen is involved in the peptide bond. So the net charge would be -2 if it's at the C terminus. And this is neutral. Okay. So then alpha helices are stabilized by what type of bonds and what are their key properties? So alpha helices are stabilized by hydrogen bonds between the alpha amino so you have N, C, I can't remember how to draw it, but anyway, there's a hydrogen bond between the residue that's 4 residues further along, okay, and it's going to be a hydrogen bond between the alpha amino group and the alpha carboxyl group, okay, and these two are going to be 4 residues apart, and so the key properties is there's 3.6 residues per turn. There's 5, and each turn is 5.4 angstroms apart, and it's a right-handed helix, and the side chains are going to point outwards. Okay. And so beta sheets are also going to be stabilized by hydrogen bonds, and it's also going to be between the alpha amino and the alpha carboxyl groups. But the difference is that the hydrogen bonds are going to be between adjacent beta strands. And so a beta sheet contains 2 to 10 beta strands. So if you look down, if you're looking down on a beta sheet, there's going to be hydrogen bonds between the strands. So this is one beta strand, and this is another beta strand, and then these two together form a beta sheet. Okay, and you can have up to 10. So you can have 2 to 10 beta strands making up one beta sheet. Okay, and they also have a right-handed turn. Okay, and they're going to alternate polar and non-polar. Okay, so what that means is that this side chain is going to be polar, and the next one is going to be non-polar. And because it's in the trans configuration, they're going to point in the opposite way. So all of the polar amino acids are going to points downwards, and all of the non-polar side chains are going to point upwards. So then you'll have one side of the beta sheet being polar and one side of the beta sheet being non-polar. Number 7, does myoglobin have quaternary structure? So what's the answer to that? No. So myoglobin is only made up of one subunit or one polypeptide chain. So quaternary structure is only possible if you have multiple subunits, because it's the way that all of the subunits interact. And it can only bind one oxygen because it's a monomer. Okay, so make sure you don't get confused about that. Why does hemoglobin display a sigmoidal oxygen dissociation curve? So the reason for having a sigmoidal curve is because it's got positive cooperativity. Okay, whenever you've got a sigmoidal curve, it means you've got cooperativity. And it means that binding of the first oxygen increases the binding affinity of the other subunits. Okay, and what chemical factors affect the oxygen binding affinity of hemoglobin? So the first one's BPG. BPG is going to bind to the center of the tetramer and stabilize the T state. And remember, T state's the low oxygen affinity state. And then carbon dioxide, or CO2. So CO2 will bind to the N terminus of the globin subunit and stabilize the T state as well. And protons, so H+. So protons can bind to the certain side chains, and it will stabilize the T state. Oh, just like side chains, like so you could have a side chain that's like let's say histidine, a histidine side chain. It can be protonated, yeah, and then so it'll pick up a positive charge, and that'll alter the shape. And then, so number 10, is there any difference in BPG binding affinity between adult hemoglobin and fetal hemoglobin, and why? So fetal hemoglobin has a lower affinity for BPG, and because BPG stabilizes the T state, it means that fetal hemoglobin is more likely to be in the R state, and if it's in the R state, it's going to have higher oxygen affinity. So if you want oxygen to go from maternal blood to fetal blood, the fetal hemoglobin has to have higher oxygen affinity. Okay, and then in the test, you're going to have to remember this equation, because they're not going to give it to you, and they're going to give you questions that where you have to use this equation. So this is the Michaelis-Menten equation. So you've got V is equal to V max, or you could write VO. So the observed velocity is equal to V max multiplied by the substrate concentration divided by Km plus the substrate concentration. So make sure you guys remember that. And so number 12, if an enzyme has a Km of 3 millimolar and the concentration of substrate and the reaction is 6 millimolar, what's the reaction rate as a proportion of Vmax? So you've got Km equal to 3, and you've got the substrate concentration equal to 6. So you're going to just put that in your equation. So that's Vmax times the substrate concentration, which is 6, divided by Km, which is 3, plus the substrate concentration, which is 6. So that's just 6 times Vmax over 9. 6 over 9 is the same as 2/3. So that's 2/3 times 2/3 of Vmax. Okay? Number 13, what's the substrate concentration when the observed velocity is equal to the maximum velocity? So the substrate concentration is equal to infinity when the observed velocity is equal to Vmax. Okay, so remember that Vmax is the maximum velocity you can get when you have as much substrate as possible. So you can just say that the substrate concentration is infinite, and that's going to come in useful when you encounter the next question. So if the equation of a Lineweaver-Burke plot is 1 over VO, so it's 1 over VO is equal to 0.6 over the substrate concentration plus 0.8, and they ask you what the Vmax is. Remember that when the velocity is maximum, when you've got Vmax, the substrate concentration is going to be infinity. So then if you divide anything by infinity, what's the answer? Yeah, so if you divide 0.6 by infinity, it's just going to be 0. So it's the same as 0 plus 0.8. Okay, so what that means is 1 over VO is equal to 0.8, and so then VO is equal to 1 over 0.8. And remember that when substrate concentration is infinity, the VO is equal to the Vmax, so that means that this is going to be your Vmax. Okay? One more question. When the substrate concentration is equal to 0.3 millimoles a liter, and the enzyme is operating at 10% of its maximum rate, what is the Km value? So for this one, we know that the enzyme is operating at 10% of its maximum rate, so that means that so what we can do is we can just say, let's say Vmax is equal to 1, okay? So if the maximum velocity is 1 and the enzyme is going at 10% of its maximum rate, then the velocity of the reaction that we can observe is going to be 0.1. Okay, so we've got substrate concentration, we've got VO, and we've got Vmax. So we just want to find out what Km is now. So we're just going to chuck the numbers in. So VO is equal to 0.1. Vmax is equal to 1. times the substrate concentration, which is 0.3, divided by the Km, which we're trying to find out, plus 0.3. So then you can just swap these two. So Km plus 0.3 is equal to 1. So 1 times 0.3 is just 0.3 divided by 0.1. And then, so you're going to minus 0.3 from both sides. So Km is equal to 0.3 divided by 0.1 minus 0.3. And that's, so 0.3 divided by 0.1 is 3. 3 minus 0.3 is equal to 2.7. Okay. So we just went a little bit over time, but hopefully that was helpful. And good luck for the test. And yeah, just feel free to message me if you guys need any help with anything. Okay? Or, yeah. Okay. Thanks.
Thank you.
You're welcome.
With the promoter,
Yeah.
when you're trying to get it into like the specific cells, so milk cells,
Yep.
how does that work with the promoter? Do you have like a bacterial promoter and then a milk